The nervous system is composed of two main types of cells, neurons and neuroglial cells. Together, these cells form the structural and functional basis of all neural processes, from reflexes and motor control to cognition and emotion. Each type of cell plays a specialized role in maintaining the integrity, communication, and support systems of the nervous system. Let's begin with neurons. Neurons, also known as nerve cells, are the primary signaling units of the nervous system. They are highly specialized for the generation, conduction, and transmission of electrical impulses. A typical neuron consists of three main parts: the cell body, dendrites, and axon. The cell body, or soma, contains the nucleus and most of the cytoplasm. It houses essential organelles such as the rough endoplasmic reticulum, often referred to in neurons as Nissl substance or Nissl bodies. These structures are involved in the synthesis of proteins necessary for neurotransmitter production and maintenance of cellular structures. Extending from the cell body are multiple dendrites, which are short, branched processes that receive input from other neurons. Dendrites carry electrical signals toward the cell body. Their extensive branching increases the receptive surface area, allowing neurons to receive input from thousands of other cells. In contrast, the axon is a long, singular process that carries impulses away from the cell body toward other neurons, muscles, or glands. The axon originates from a specialized region of the soma called the axon hillock, which plays a critical role in initiating action potentials. Many axons are covered with a myelin sheath, which increases conduction velocity. The axon may branch at its terminal end into structures called axon terminals or synaptic boutons, which form synapses with target cells. Neurons can be classified in several ways. Functionally, they are categorized as sensory, afferent, neurons, motor, efferent, neurons, and interneurons. Structurally, neurons are described as unipolar, bipolar, or multipolar based on the number of processes extending from the soma. Most neurons in the CNS are multipolar, while pseudounipolar neurons are typical in sensory ganglia of the PNs. Neurons are postmitotic, meaning they do not divide after full maturation. This makes their maintenance and protection vital, especially in response to injury or disease. Now let's discuss neuroglial cells, commonly referred to as glia. Neuroglia are non-neuronal support cells that vastly outnumber neurons in the nervous system. Although they do not conduct electrical impulses, they are essential for maintaining homeostasis, providing support and protection, regulating the extracellular environment, and participating in immune defense and synaptic modulation. In the central nervous system, there are four major types of glial cells. First, astrocytes. These are star-shaped cells with numerous processes. They maintain the blood-brain barrier, regulate ion concentrations in the extracellular fluid, recycle neurotransmitters, and provide metabolic support to neurons. Astrocytes also participate in repair processes and form scar tissue after CNS injury. Second, oligodendrocytes. These cells are responsible for myelinating axons in the CNS. A single oligodendrocyte can extend processes to multiple axons, wrapping segments with myelin. Myelin is a lipid-rich membrane that insulates axons and increases the speed of impulse conduction via saltatory conduction. Third, microglia. These are the resident immune cells of the CNS, derived from mesodermal precursors. Microglia constantly survey the CNS environment for damage or infection. Upon activation, they transform into phagocytic cells, engulfing pathogens, dead neurons, and cellular debris. Fourth, ependymal cells. These ciliated, epithelial-like cells line the ventricles of the brain and the central canal of the spinal cord. They help produce and circulate cerebrospinal fluid, CSF, contributing to the homeostasis of the internal CNS environment. In the peripheral nervous system, there are two primary types of glial cells. First, Schwann cells. These cells form the myelin sheath around peripheral axons. Unlike oligodendrocytes, each Schwann cell myelinates only one segment of one axon. They also assist in the regeneration of damaged peripheral nerves by forming regeneration tubes that guide axon sprouting. Second, satellite cells. These cells surround the neuronal cell bodies in peripheral ganglia. They provide structural support and help regulate the microenvironment around sensory and autonomic neurons. Neuroglial cells are capable of cell division throughout life, unlike neurons. This regenerative potential, however, also makes glial cells the origin of most CNS tumors. For instance, astrocytomas and oligodendrogliomas arise from astrocytes and oligodendrocytes, respectively. The interaction between neurons and glial cells is dynamic and critical. Astrocytes and oligodendrocytes regulate synaptic activity and plasticity. Schwann cells facilitate axonal regeneration. Microglia participate in synaptic pruning during development. Glial dysfunction is now recognized in a wide range of neurological disorders, including multiple sclerosis, Alzheimer's disease, epilepsy, and gliomas. In summary, neurons are the excitable, information-transmitting cells of the nervous system, while neuroglia serve vital support, protective, regulatory, and homeostatic roles. The intricate partnership between these cell types allows the nervous system to function efficiently, adapt to stimuli, repair itself, and maintain overall neural integrity.